The DNA alphabet, the natural language of life. The DNA alphabet is a fascinating concept that serves as a bridge between molecular biology, linguistics, and the general curiosity about life's building blocks. This paper aims to provide an easy-to-understand overview of the DNA alphabet, its components, and its significance. We will also delve into some theoretical ideas that provoke thought about future applications and research in this area. DNA, the molecule that carries the genetic instructions for the development, functioning, and reproduction of all known living organisms, has often been compared to a language. This paper is inspired by Edwards' video on the DNA alphabet, which serves as an approachable introduction to the subject. The DNA alphabet refers to the 20 amino acids, selenocysteine, and the stop command that make up the language of life. In DNA, the sequence of nucleotides, A, T, C, G, forms the basis for the language of life. These sequences translate into amino acids, which are the building blocks of proteins. The order of these amino acids is crucial, much like the order of letters in a word. Each amino acid is represented by a specific sequence of nucleotides. For example, the amino acid lysine is represented by the sequence AAA or AAG. These sequences serve as letters in the DNA alphabet. Think of DNA like a recipe book for making you. Just like how recipes use a combination of 26 letters in the alphabet to form words and sentences, your DNA uses a combination of 22 letters to form the instructions for your body. These letters are actually amino acids and special commands that tell your body how to function. Lysine, asparagine, isoleucine, etc. These are like the ingredients in a recipe. They are the building blocks that your body uses to make proteins, which are essential for everything from moving your muscles to fighting off infections. Methionine or start, this is like the word begin at the start of a recipe. It tells the body where to start reading the DNA instructions for making a particular protein. Stop and selenocysteine, these are like the words end or stop at the end of a recipe. They tell the body when it's done making a particular protein. Understanding this alphabet can be useful for various applications. Medical research, knowing which letters correspond to which functions can help us understand diseases and how to treat them. Genetic engineering, if you know the alphabet, you can theoretically edit the spelling to create organisms with specific traits. Personalized medicine, understanding your own DNA alphabet can lead to treatments tailored specifically for you. Lysine, important for protein synthesis. Asparagine, plays a role in the biosynthesis of proteins. Isoleucine, essential for optimal growth. Methionine or start, initiates the translation process. Arginine, necessary for the synthesis of proteins. Serine, involved in metabolism. Threonine, important for the nervous system. Stop and selenocysteine, signals the end of translation. Tyrosine, precursor to neurotransmitters. Leucine, stimulates muscle protein synthesis. Phenylalanine, precursor to tyrosine. Cysteine, contributes to protein structure. Tryptophan, precursor to serotonin. Glutamic acid, important neurotransmitter. Aspartic acid, involved in the citric acid cycle. Valine, essential amino acid. Glycine, simplest amino acid. Alanine, important for energy. Glutamine, involved in immune function. Histidine, precursor to histamine. Proline, important for protein structure. Imagine a language so intricate that it forms the blueprint of life itself. DNA, with its complex sequences, resembles an encrypted alphabet. By probing into the depths of genetic makeup, we are faced with the tantalizing possibility: could the DNA alphabet transcend earthly boundaries to be a cosmic language? If such a language is universal, it has profound implications, including the potential to decode extraterrestrial life forms and to truly grasp the interconnectedness of all living things. The search for this universal language could revolutionize our understanding of life's diversity and unity. In the digital era, where data is as precious as currency, DNA emerges as a formidable candidate for the ultimate data storage medium. Its capacity to hold voluminous information in a minuscule space is unmatched. we stand on the brink of a data revolution, exploring how strands of DNA can be synthesized and sequenced to store everything from literary classics to entire databases. Such a technology could lead to data longevity that eclipses current storage methods, preserving knowledge for millennia. For years, a significant portion of human DNA was dismissed as junk, without purpose or function. However, emerging hypotheses suggest that these enigmatic sequences may be a hidden treasure trove of biological potential. Imagine if we could harness high-frequency vibrations or other stimuli to wake up these dormant segments. The implications are vast and varied, ranging from revolutionary gene therapies that could eradicate hereditary diseases to the evolution of new human capabilities. This could be the key to unlocking true human potential and catalyzing an evolutionary leap forward. These exploratory avenues not only promise to broaden the horizons of scientific knowledge but also challenge our fundamental perceptions of life, identity, and our place in the cosmos. Delving into these questions could yield transformative insights, leading humanity to a future where we are not just observers of the genetic code but active participants in its orchestration. The DNA alphabet serves as a foundational concept for understanding the complexity and beauty of life. Its 22 letters provide the instructions for creating and sustaining life, and its study opens up a plethora of theoretical and practical applications. Further research in this area is not only exciting but essential for unlocking the secrets of our existence. According to Schaff-Brady, the DNA alphabet could be simplified to A plus A. This could revolutionize our understanding of genetics by making it easier to read and interpret DNA sequences. This simplified alphabet could potentially eliminate the concept of junk DNA, as every letter and its variation would have a specific, known function. A simplified DNA alphabet could make it easier to use DNA for storing digital data, a method that could be far more compact and long-lasting than current technologies. A simplified, well-understood alphabet could accelerate advancements in gene therapy, making it easier to correct genetic diseases. If proven to be more accurate, this simplified alphabet could serve as a universal genetic language, aiding in research collaboration and even the search for extraterrestrial life. Understanding and simplifying the DNA alphabet could lead to the eradication of genetic disorders, as we could more easily edit faulty letters. By simplifying and fully understanding the DNA alphabet, we open the door to incredible advancements in medicine, technology, and even our fundamental understanding of life itself. In my considered opinion, the current DNA alphabet, while intricate, is not capturing the full scope of genetic complexity and potential. I believe that there are missing elements in our understanding of DNA, elements that could unlock new dimensions of biological function, medical treatment, and even human evolution. While the existing system of 22 letters, comprising 20 amino acids, selenocysteine, and the stop command, has served us well in decoding the language of life, it may not be the final word. My proposition of an alternative DNA alphabet, using A plus A, may seem to simplify the system at first glance. However, I argue that this new format could actually reveal complexities not yet understood, acting as a missing key to a more advanced understanding of genetics. This is not merely a matter of semantics or nomenclature, it's about the potential to revolutionize our understanding of everything from basic cellular function to the treatment of complex diseases. While critics may argue that simplifying the DNA alphabet could lead to a loss of nuance and biological detail, I counter that the missing key could actually provide a more accurate and comprehensive understanding of genetic function. It could be the paradigm shift needed to move from a standard model of genetics to a unified theory, much like in physics. The implications of this could be monumental. Imagine more effective gene therapies, more nuanced understandings of genetic diseases, and more targeted medical treatments. On a broader scale, this could lead to advancements in biotechnology that we can't yet even conceive. In summary, while the existing DNA alphabet has brought us far, I believe that a new, more advanced system could take us even further. It's time to explore what we might be missing and to consider that the keys to the next frontier in genetics may already be within our grasp. For more information on the ideas presented here, visit edward-raby.com by Schaff-Brady, Nottingham, United Kingdom. Like this video and subscribe to our YouTube channel for more free videos. Check out researchforum.online for the full research paper.